accolades, but it's a pleasure to introduce Ben. Just as a brief history, he received 2 bachelor's degrees. One wasn't enough, you had to get 2. One in chemistry and one in biology from University of North Carolina. And then he worked for as an analytical chemist for a pharmaceutical company for 3 years. And so he's obviously well-versed in chemistry. And then he came here to get a PhD and came to work in our department, in our program. And I had a project on the ecology of the shelf environment off of Cape Canaveral. And he joined that effort and actually led the phytoplankton component of that study. And it's interesting because the phytoplankton community structure of that area is very poorly understood. With the exception of satellite imagery, there's really very little information on the community structure of communities along the shelf of the east coast of Florida until you get down further south in the Miami area, Key Biscayne, and the Florida Keys. So I think the data and results that Ben generated and is currently, we're working on some publications to get out, is actually going to serve the community very well in terms of a better understanding of that system. So with that, I will give it over to Ben. Take it away, Ben.
All right, well, thank you, Dr. Flips. Like you said, my project was centered around Cape Canaveral, Florida on the inner shelf. And I looked mostly at seasonal phytoplankton biomass and composition. I won't discuss too much composition today because I don't think too many people will really know, what a Gyrodinium spirale is and things like that. But I will talk mostly about functional groups and their differences in the surface and near-bottom water and how they change seasonally. So the objectives of the study were to compare seasonal trends in surface phytoplankton biomass. This was the first objective. And then we used 3 separate measures for that. So we had extracted chlorophyll that we took at sites. We had satellite chlorophyll estimates that we used Google Earth Engine and the MODIS satellite for. And then we had biovolume derived biomass, which we used microscopic counts to derive. And then we compared the biomass of different phytoplankton functional groups along depth and seasonal gradients. And then we also were interested in seeing how those groups and overall phytoplankton biomass will change by environmental drivers and how that affected abundance and composition. So just a brief introduction for people who aren't as well versed in phytoplankton. Phytoplankton, or sometimes referred to as marine protist, even though protist doesn't necessarily include cyanobacteria, which are a large part of the phytoplankton group. They control many biogeochemical cycles across the globe and are responsible for half the primary production. They can vary in size from a 1 centimeter picocyanobacteria all the way up to a couple hundred micrometer centric diatom. And so that can be 7 orders of magnitude in biovolume. There's over 150,000 species found using DNA sequencing studies, but only around 11,000 have been seen or described. So there's still a wide breadth of diversity. And then they can have a diversity of lifestyles. Phytoplankton generally are seen as photoautotrophic, and so they do primary production through photosynthesis. But dinoflagellates and small naked flagellates can live heterotrophic lifestyles and even mixotrophic lifestyles where they can adapt whether they use photosynthesis or capture prey or capture large molecules for their carbon acquisition. So first of our functional groups that we talk about in our study is diatoms, and they come in a wide range of sizes, but they are autotrophic, and they tend to have a large growth rate, especially when they're smaller. And so they have a preference for nitrate and and other inorganic forms of nitrogens, and they can respond to it very quickly. So they are found in high abundance, especially in upwelling areas. And And then also we have Cyanobacteria. They come in many forms. There's colonial forms like here seen on the upper right. There's filamentous forms, as you can see throughout the figure. And then they even have picocyano forms that are more solitary, that are much smaller and aren't colonial at all. For our study, the Cyanobacteria included both of these forms, both filamentous and the small spherical pico-sized photoautotrophs. So they tend to dominate when nutrients are limited. And this is because many of them can fix their own nitrogen. And so they don't need nitrogen from an outside source. And then lastly, of the main functional groups that we'll talk about are the dinoflagellates. Here's a nice figure of some armored dinoflagellates, but they can also be naked dinoflagellates like Corenia brevis off the west coast of Florida. And they tend to be larger, slower growing, but they have the advantage of having more flexibility in their trophic models. So they can, many of them are mixotrophs. Some of them are obligate autotrophs, but many are mixotrophs. And then there are some that are heterotrophs as well. So they are flexible across all of those, and they can acquire nitrogen from a variety of sources. And then our catch-all group, which we kind of made the other group, which was mostly nanoflagellates. And these are similar to dinoflagellates, and they, it's not necessarily a grouping of bitrophic model because they're just really difficult to look at under the microscope. So they can have trophic modes of heterotrophy, mixotrophy, phototrophy, and they can eat bacteria. But as a group, it's more a function of necessity. So across Florida, there's been many studies on the more eutrophic regions of Florida. And this includes the western shelf and the Indian River Lagoon, which Dr. Flips here spent much of his career looking at. But there's very little known about the Atlantic shelf of Florida, which is just outside the Indian River Lagoon, but it tends to be much more oligotrophic. There is not as much nutrient input from human sources. So it is not as well studied, mostly because there hasn't been many eutrophic issues in this region. And so for us, we had many goals for this study. We wanted to add more rigorous data on the phytoplankton abundance in this region outside of satellite data. And then we wanted also to add some species and morphological data. And along with that, this is a long 6-year data set. So it'll be a nice, robust data set for someone who's looking at this region, maybe looking at invertebrates or fish, or just looking at biogeochemical cycles or anything like that. So further site description, here's a nice figure looking at the intensity of the Gulf Stream seasonally. And while the Gulf Stream does intensify during the summer, surface eddies and does not interact a ton in the summertime, but it can at times travel inshore and bring warm surface water from the Gulf Stream into our study region. And then looking at our sampling sites, here you can see the red arrows with the 3 main regions that we studied. And if you look at the upper right of the figure, you can see the drop off of the shelf, and this gradient in depths may allow for some seasonal upwelling that we will discuss later. And hold on. But a better look at our sampling regions. So there's 3 regions, and they're each sandy shoals. So there's a ridge and a swale, and it's Bull, Chester, and Cape Canaveral shoals. And then we separated the shoal into the North Shoal and South Shoal. The blue side, or the blue region, is the ridge, which would be the top of the shoal. And then the green is the lower half of the shoal, the swale. And this is a shallow inner shelf, so its depth is only from around 3 meters to 20 meters. So it'd be around 3 meters at the top of the shoal, down to 20 in the swale regions. So it's rather shallow. And then the sampling design was coincided with other species and other studies. So it wasn't necessarily designed to study phytoplankton, but also invertebrates, fish, and there were some oceanographic studies as well. And for methodology, the sampling protocol, just talk about it a little bit more. 24 sites were sampled for each date every season from the fall of 2013 to the summer of 2019. There was a surface sample, which was a 2.5 meter integrated pole that we, so we didn't just skim the surface, but we used an integrated pole. And then a bottom sample was used with a Niskin bottle, which is kind of shaped like a football, an empty football that we can throw down and get close to the sediment bed, but we aim to be around a meter off the sediment bed. So it's not a true bottom sample, but more of a near bottom sample. And then for each of these samples, we had a micro and nanoplankton, which was preserved in lugol's. We had an autotrophic picoplankton, which was preserved in glutaraldehyde, and then a chlorophyll and nutrient sample. And for phytoplankton biomass, here's a nice little flow chart to look at. For biomass, we wanted to look at multiple different values. So we had chlorophyll, which we sampled and measured spectrophotometrically, and then a satellite data that we got from Google Earth Engine. And then along with that, we had biovolume data that we converted to carbon biomass. And that was collected using the microscope. So we compared all of these. And so for the two chlorophyll biomasses, we compared those to see if there was a relative agreement, even though satellite has much higher resolution, both in time but much larger space. We wanted to see if our samples collected a pretty representative seasonal idea of what was going on in the region. And then we also wanted to compare chlorophyll with the biomass derived from biovolume to see if there were some hiccups in the biomass that you would get looking at chlorophyll data versus looking at actual phytoplankton sizes and measurements. So just how we got some of these chlorophyll, we measured the optical density using a spectrophotometer using the American Public Health Association's 1998 protocol. And then total phosphorus and total nitrogen were determined by the Soil and Water Science Analytical Lab. And then water temperature and salinity were recorded on site using a YSI field probe. And we measured both at 0.5 meters for the surface sample. And then for the near bottom sample, we aimed for around 1 meter off the bottom. For the satellite chlorophyll, we used the Modus Aqua satellite chlorophyll A product using Google Earth Engine. And then we used a four-sided polygon that we lined up the latitude, longitude, and took an average of each pixel inside the region, and that was one chlorophyll point. And then for the monthly average of chlorophyll values, we averaged all those, the cloudless days to get a monthly value. And so we tried to do it based on month, since our data was seasonal. So we didn't think a daily resolution was needed. And then one caveat to this data was it is an algorithm that was intended for open ocean oligotrophic water. So there may be some issues with turbidity and other surface reflectance that wouldn't be an issue in open water that might be an issue for our coastal region. And for the microscopy, we looked at both picoplankton and phytoplankton. The picoplankton was at 1000x using epifluorescence, and then the phytoplankton was counted using an inverted microscope. For phytoplankton less than 30 microns, we looked at them at 400x and did 100 grids. And for phytoplankton larger than 30 microns, we looked at the whole bottom sample, and this was made using a settling chamber. The cells per mil were calculated using this equation, and then biovolumes were estimated from geometric shapes of plankton, and then from that, the biomass was calculated. And then for statistical analyses, we did many biomass mean comparisons along chlorophyll, functional group biomass, and then we looked at different seasons and depths as our main comparison. And we used the Games Howell post hoc test for these comparisons, mostly for unequal variance, because as the biomass goes up, the variance tended to go up as well. So the equal variance assumption could not be met. So the Games Howell did a good job of helping with that. And then for multivariate analysis, we used species abundance data and environmental parameters. And then we did a transformation-based redundancy analysis. And just to talk a little bit more about that, because the raw counts for phytoplankton especially are really difficult to use, given the wide differences in size. And so a lot of people have come up with ways to help with that. But the way I found most appropriate was to use biovolume data as for abundance rather than count data. And that tend to regulate. So if you have 1,000 or 10,000 cyanobacteria and one large centric diatom, the biovolume data would kind of equal the abundance to, or normalize the abundance so that, you know, one isn't necessarily greater than the other. And since there were so many species that we saw in our data set, like I think we saw over 500 or 600 species, but many of them had very, very low abundance, maybe seen in a couple of sites in a couple of seasons. We for this analysis, we took the top 100 abundant groups or abundant species genera, and those were used for analysis because we had more regular data on them. And then we normalized the environmental explanatory variables, usually using a logarithm transformation. And then we performed a forward selection of those variables. So the RDA is pretty much a multiple regression, but it also has multiple dependent variables rather than just one dependent variable like you would have in a normal linear regression. And so for results, the chlorophyll for the satellite, we saw a fairly regular seasonal pattern with a large surface chlorophyll values happening in the fall and winter time, and especially in the last 2 years where we saw large spikes in chlorophyll and in early 2018 and early 2019. This same trend was seen with our sampled values, which was good, but we did the peak for 2019 was not quite as large as the peak for the surface values of the satellite. So it goes to show that the satellite with its temporal resolution really picks up on, you know, some short-term large phytoplankton biomass that our sampling wasn't able to necessarily pick up on, but we did get the same seasonal trend. And here with the box plot, looking at the box plot, we can see that June was the month with the lowest chlorophyll values on the surface, with almost every June and summer sampling period having very low surface values. And here's just a graphical comparison with the sampled chlorophyll versus satellite chlorophyll. overall, the satellite chlorophyll values were higher, as you can see by the most of the values lying under the 1-to-1 line, which is here in with the red dashes. And especially when the satellite chlorophyll was around 4 micrograms per liter or more, we did not see a good comparison between that and the sampled chlorophyll values. And this could be due to the satellite maybe not penetrating far enough into the water column, or maybe there's some turbidity in the surface water that the satellite algorithm is interpreting as chlorophyll. And now more of a 1-to-1 comparison between our chlorophyll concentrations and our biomass that we derived from the microscope. They matched up pretty well overall. There were some outliers, like our first sampling period when we had a lot of dinoflagellates and some filamentous cyanobacteria. We did not see a large concentration in chlorophyll, but we did see a large concentration in phytoplankton, and this was, in filamentous bacteria, cyanobacteria, and dinoflagellates, excuse me. And we also saw a difference here in June of 2015 and December of 2014, where we saw much higher chlorophyll concentrations than we did biomass. And this could be due to higher chlorophyll concentrations inside of the cells, or it could be just some error in the data. But overall, our chlorophyll and carbon biomass values tended to agree rather well, and the variance was spread both above and beyond, or above and under the line. The one exception is here in the fall of 2013. These two values are from, which we discussed, is like because of the dinoflagellates and cyanobacteria, and it's most likely because dinoflagellates may be heterotrophic, so they might not have as much chlorophyll in their cells. Now, the seasonal comparison of the functional groups, diatoms were much higher in the spring and winter. This is not accounting for any depth variables. So diatoms were about the same in the spring and winter, not accounting for depth. Dinoflagellates showed a much higher preference for the fall, and cyanobacteria showed a higher preference for the summer, and the fall were pretty close, but they were statistically significantly different from each other, but the summer showed the highest cyanobacteria biomass. And then the nanoflagellates or the naked flagellates, they showed a preference for the fall as well, similar to the dinoflagellates. And then for the spring and winter, they showed a similar higher concentrations in diatoms, higher concentrations in cyanobacteria. And the summer was dominated by cyanobacteria. And then the fall showed the highest variety of high biomass with cyanobacteria, dinoflagellates, all having their one of their highest biomasses in the fall, with the diatoms also not having their lowest in the fall, but they were not as dominant as the other groups. And then now looking at depths rather than seasons. So diatoms had a strong preference for the near-bottom samples, both for ridge and swale sites. The swale sites had the highest biomass. Dinoflagellates showed a preference for surface, and cyanobacteria and the other flagellates group showed no preference based on the surface or bottom water. And here, just looking at the surface overall, they both showed the same pattern, with cyanobacteria being the highest in the surface overall, and followed by dinoflagellates. And then the bottom was dominated also by cyanobacteria and diatoms. Putting it all together, and this is the large table, I won't try to press too much, but so diatoms in the spring showed much higher biomass. Spring and winter showed much higher biomass in the bottom region, and they dominated these two periods. Dinoflagellates actually did not show a surface preference for any of the seasons when added in with season. That this surface preference was only seen in the fall in great numbers. And the other category, the flagellate category, showed a surface preference in the fall, but did not show the surface preference in the spring, and actually were higher or showed higher abundance in the bottom water in the spring. So it's kind of an interesting pattern. And now comparing these functional groups to physical parameters, try to focus on the other flagellate group positively correlated with dinoflagellates and diatoms, but dinoflagellates and diatoms did not correlate at all with cyanobacteria. And cyanobacteria did not correlate with the flagellate group at all either. The surface groups tended to be negatively correlated with all these physical parameters that were associated with summer, other than cyanobacteria, which showed no correlation. And then cyanobacteria was correlated with total nitrogen, and so was dinoflagellates, maybe showing a pattern between those two groups in the surface. In the bottom, the total nitrogen was also correlated with cyanobacteria, showing no difference from the surface. But the dinoflagellates showed no preference for bottom, even with higher total nitrogen values. And diatoms were positively correlated with total phosphorus in the bottom water. And then once again, the other flagellates groups was corresponded with diatoms and dinoflagellates, possibly showing their dual role as, as a group. They can be made up of mixotrophic species and phototrophic species. And then one last figure here is the species and physical parameter multivariate analysis. It kind of just reiterates some of the patterns we saw in the other tables, with this blue circle showing many of the dinoflagellate species appearing most in the fall. And then the picocyanos, which are kind of hard to see because they're behind the temperature label, but the picocyanos and the Synechococcus, which is under the summer label, are both highly correlated with summer, both in temperature and as a season, and with precipitation, salinity, things like that. And then diatoms showed a much wider breadth of abundance depending on species composition. They were seen with high TP values in the bottom water as we saw, and then they correlated in the spring and winter time. And this RDA analysis overall explained 50% of the variance that was explainable by the variables in this graph. And here's just some notable biomass events. I discussed a little bit the fall of 2013. There was a large trichodesmium bloom, and it was associated with high abundances and diversity of dinoflagellates, and also had a high total nitrogen. And that was probably due because trichodesmium does fix nitrogen. And this association has been seen on the west coast of Florida as well with Correnia brevis. And then in the spring of 2018, there was the highest diatom biomass that was seen during our entire samplings, but there was no notable elevated TP or TN. And then in the winter of 2019, there was high diatom biomass that was associated with elevated TP. And then just a brief discussion. This region, a study by Eutrophic in 2019, which correlated with our samplings, showed a distinct upwelling events happening in the summertime, as you can see here from July, August, and September. The water temperatures seem to decrease when they should be increasing with the higher light irradiance and temperatures. So this is an interesting pattern that's seen here, and I'm not sure how much of this is reflected in the phytoplankton species, because this region has a really low resonance time and the water moves around quickly. But we did see this pattern definitely in the summer of 2016, where we noticed an almost 6-degree difference in the bottom and surface temperatures, where and like I said, this region is very shallow, so this temperature difference would not be expected in the summer without some sort of cold water input from off the shelf. And as you can see in 2019, which I don't have an arrow for, but in 2019 summer, the difference in water temperature in the surface and bottom is almost non-existent. And then just a brief summary. So the surface biomass was highest in the fall. That's when a lot of the phytoplankton groups showed the highest biomass. And then dinoflagellates had a distinct preference for the surface during this time. And then the near bottom biomass was dominated by diatoms and cyanobacteria in the spring and winter especially. And then the summer was dominated clearly by cyanobacteria. None of the other groups showed a high biomass during this time. And flagellates showed a varied preference for the fall surface and then the spring bottom. So as a group, they were a little bit more difficult to make some conclusions from. So surface nutrients in this region were depleted much of the year, and explains why cyanobacteria was such a high and prevalent group. And this included diazotrophs, which is another name for nitrogen fixing cyanobacteria. And these could be possibly a catalyst for dinoflagellate growth. We really only had one sampling period where we saw a lot of the trichodesmium and dinoflagellates. So it's kind of hard to make a conclusion from that, but this pattern has been seen in other studies. And then terrestrial sources of nutrients is also kind of difficult to understand where it's coming from. We did see upwelling in the cold water, but we're not sure what terrestrial sources may add nutrients into the system, and where the nutrient, or if the nutrients might be coming from, possibly some upwelling or even from along shore currents from the north. So we're currently working to try to figure that out. And then, like I said again, the water residence times here are really short because it is offshore and there's no barrier islands or anything like that. So the water is constantly moving. And so much of the phytoplankton that we're seeing may be coming from either the south or the north, and may not originate in the region that we're studying. So we may be seeing patterns that are just moving through. Future directions, it'd be interesting to see the extent of near bottom diatoms that are seen in this region, and whether they're responding to coastal upwelling or if they're just travelers bypassing the region. And then what the extent of grazing of nanoflagellates and dinoflagellates are on these trichodesmium blooms that do appear in the oligotrophic Atlantic often. And then it's the niche partitioning the phytoplankton groups in the fall season was very interesting, and what may be a effect of other zooplankton besides dinoflagellates during this time when the surface biomass of all these both protists and cyanobacteria are high. And then just a couple of methods that may help with this is the imaging flow cytobot. This is the phytoplankton dream, if you will, where there's a machine that is looking and giving you good images of phytoplankton that you can process and look at as a taxonomist, so you can spend less time on the microscope. And then of course, molecular methods are really common in this field, and they help a lot with the pico and nanoflagellate diversity. And this field is just opening up, where this diversity is starting to be uncovered. And then I'd like to thank Dr. Phillips and the rest of my committee members, all the BOEM associated phytoplankton collectors and counters and researchers. This includes Susan, Leslie, Hama-chan, and Macy, who was a master's student, Paula, Jean, and then Dr. Muri, and all the other people on the BOEM project. I'd like to thank Karen Bray, the SNRE champion. She keeps me in line, sends me multiple emails to remind me of things. And then of course, I'd like to thank SNRE, SFFGS, and BOEM for allowing me to be here. So thank you.